There were 6 people in the study that you presented this morning, and safety was the primary endpoint here, but we were also hoping for some hint of efficacy.
Yeah.
Is that true?
Well, there are three things we wanted to accomplish. First, we wanted to show that logistically we could send patients to an apheresis unit, get their white cells, send them to a manufacturing plant, have those cells enriched for CD4, transduced with the gene therapy, expanded up to 10 to 30 billion, sort of clinically relevant scale, frozen, shipped back to the clinical site, and be able to thaw and give those cells back to patients. So we've shown that we can complete that loop.
Right.
Number two, primary issue is is it safe? Is it
Yeah.
safe and well tolerated? I think the answer is unequivocally yes.
Yeah.
And the third was a number of markers that we're looking for to see how successful this was. And in terms of CD4 count, in terms of CD4 CD8 ratio, the engraftment and expansion of those cells, the persistence of those cells, and the distribution of those cells into the gut mucosa. And all five counts, I think the study was a raging success.
Yeah, I mean, from all the studies that I'm aware of where they did any kind of CD4 expansion and modification, this is orders of magnitude more
Orders of
successful.
magnitude better. I
Yeah.
don't think the results from this study could have been any better.
Yeah.
Now, the caveat to that is that this study was done in aviremic hosts, individuals who were fully suppressed. So the modified cells have the same proliferation kinetics as unmodified cells in HIV negative, both in vitro and in vivo. In HIV positive situation where these cells have a selective growth advantage. So in that individual where there's viral replication, these cells should take off.
Yeah.
But in the study that we did, I don't think these results could have been any better. And they do set a sort of new landmark for cell therapy uptake.
Sure.
But we have to remember that this could all amount to nothing if these cells, either peripheral blood cells or stem cells, have to be able to generate HIV-specific effector cells that can reduce viral load. And until we demonstrate that, we've accomplished very little.
Now, the study that you mentioned that's going to happen next, which is looking at people who are treatment naive, is that possibly going to give us that?
That's going to give us the answer.
Okay.
Because those folks, because they're treatment naive, have a measurable viral load. At screening and at baseline, they'll get those cells, and presumably in the setting of that viremia, we'll see a better engraftment and uptake of the cells. And that is a question of what happens to those viral loads. If those viral loads come down, then we're entering a new era of HIV care. If those viral loads don't come down, then we have to move on to stem cells. And stem cells have pros and cons. You're likely to get a fuller repertoire with a stem cell gene therapy, but to use stem cells comes with a certain risk, and that is that the conditioning agents carry the risk of leukemia. And so it's better to start with peripheral cells. They appear safer, and see if we can make this work. If we can't, we're going to have to move on to stem cells.
Last question, this may be the harder question to answer, which is, our readers are going to be so interested in, can this happen for me? Can this become a real-world solution? And, current ARV regimens range in price from $14,000 to $30,000 a year, depending on how many someone takes. Is this scalable? Is this something we might be able to do?
The answer is definitely, it is scalable. Right now, we're spending a lot of money on each patient we enroll in the clinical study. But if this works and we are having antiviral effects, then I think they're very easy to make a cost-benefit analysis in terms of, depending on what the benefit is. If there is the opportunity to discontinue antiviral therapy, it wouldn't take much for this to be cost-effective.
Great. Any closing thoughts?
Fingers crossed.
Okay, great.